Hello and welcome back to our playlist where we are solving Rosalind bioinformatic problems. My name is Marcel, and in this video we're gonna learn we can transcribe DNA into RNAs. Okay, so here we have a description about the second nucleotide, nucleic acid with the RNA. Okay, so the problem here is we must know that RNA string is a string formed by A for adenine, C for cytosine, G for guanine, and U for uracils. Okay, so given DNA string corresponding to the codon string, the RNA transcript it will be formed when we replace the T by U's. So we must remember that RNAs have uracils while DNA has thymines. So here is the problem. We're going to receive a DNA string with a length at most 1,000 nucleotides, and we should return the transcribed DNA. Okay, so let's add model this problem. Okay, we get it here, get string here, and now we're going to create a function called transcribe DNA. Okay, this function should receive a DNA string, the type string, and should return a string. And as we did here in the count nucleotide, we must guarantee that the user provide a valid DNA string. So let's use it here. We're going to copy the same code here. Okay. Now what we're going to do is to use the replace method in the DNA string. So DNA dot replace, and we're going to replace T by use. So we're going to replace all the T's that we have in DNA string by use. Okay, so we're going to replace T mines by uracils. So looks easy, right? So let's test it. So I'll define it as sick. Okay, and now I'm going to use this transcribe DNA. Okay, let's try to run this here. Okay, the script didn't return any code because the transcribe here. Let's do this. Okay, now we can. Oops, now we can run this, and this. Okay, so our function returns this sequence. Let's check it. Okay, so yeah, we got it right. Okay, so our function works, right? So we'll clean it or DNA2 here because as you remember, this is where we are writing the functions to work, functions that could work in so many other problems. Okay, so here now let's define the code, the script for solving this problem. So I will just copy the DNA. Okay, but now we don't need to import count nucleotides. We want to import transcribe DNA. Okay, and here I want to def the solve function, and the solve function will open. Let's use the alt string here in our mode as f. Oops, sorry. This we're gonna to fill with the database that we're going to download. Okay, so now we're gonna use the DNA seq. It will be f read dot stripe. So we're going to stripe lines. Okay, and the RNA-seq here will be the function transcribe DNA in the DNA-seq. Sorry, in the DNA-seq. Okay, and finally we're going to print the RNA-seq here for we get the results in the terminal. Okay, so if let's copy it here because it's basically this part is the same. Okay, so cross your fingers. Let's download this new data set here. Okay, so 1 second. I'm getting it here. So here, Rosalind number 4. So copy relative pay. Let's save it here. Now I'm going back to PowerShell terminal and let's run Python and source scripts dot RNA. Cross your fingers. Yeah, voila. Or at least we have an RNA-seq. So let's copy and paste here in the answer. And yes, we got it right. So as you can see, this is a really easy problem. We just need to use the replace method to change the T by use, and we got the transcribed RNA. Okay, so let's see what will be our next problem for our next video, but I will I'd like to ask you, if you are liking this type of videos, to subscribe in our channel, to give a like. It would be